I'd like to welcome you to this webinar on the role of phagocytes in homeostasis and inflammation. I'd also like to personally thank our sponsor, R&D Systems, a biotechnology company, for their generous support of this webinar. This year represents the 100-year anniversary of the death of Élie Metchnikoff, a Ukrainian zoologist who has been credited with the seminal discovery of the phagocyte back in 1882 at the University of Messina, and for which he was awarded the Nobel Prize in Physiology and Medicine in 1908. The study of phagocytes, which the macrophage is the canonical subset, but which also include dendritic cells and neutrophils, has experienced a rebirth in the last 15 or so years. And while traditional functions had been attributed to engulfing pathogens leading to their clearance, we've learned that these cells are much more versatile and include roles in regulation of metabolism, as well as maintaining tissue homeostasis. We're fortunate to be joined by three leading investigators in the field of macrophage and dendritic cell biology. Caetano Reis Sousa is a principal group leader at the Francis Crick Institute and professor of immunology at Imperial College in London. Caetano is studying the properties of dendritic cells that render them immunogenic upon interaction with dead cells or microbes. Ajay Chawla is a professor of physiology and medicine in the Cardiovascular Research Institute at the University of California in San Francisco. Ajay will be speaking about the role of macrophages in mitigating environmental and dietary stress to maintain metabolic homeostasis. Finally, we're joined by David Russell, the William Kaplan Professor of Infection Biology at Cornell University's Veterinary School in Ithaca, New York. David is studying the interactions of macrophages with pathogens, with the pathogen Mycobacteria tuberculosis, and he's leveraging his observations to facilitate the generation of vaccines to this pathogen. Before we get started with the talks, we have a few practical points we'd like to raise. Each of our speakers will talk for about 15 minutes, and following the talks, we'll have a question and answer session that covers all three presentations. If you have a question for any of the three speakers, just click on the question tab at the top of the display and type it in. You can enter your question at any time, and there's no need to wait until the end of all three presentations. If your query is directed to a specific investigator, please indicate that, so we'll be able to direct the question appropriately. We'll try to do our best to pass on as many of the questions as possible within the time allotted. Finally, we are grateful to all three speakers for making time to join us today, and to our sponsor, R&D Systems, for supporting this webinar. And we'll then start now with Caetano Reis Sousa. Caetano, the platform is yours.
Well, thank you very much, Bruce. So the webinar that we have today effectively celebrates the 100th anniversary of the death of Metchnikoff, which, as Bruce mentioned, is famous for proposing phagocytosis as a mechanism of host defense, thereby giving birth to the concept of innate immunity. Now, at around the same time, we also witnessed the birth of adaptive immunity with the work of Paul Ehrlich, who proposed that defense from infection could be mediated through humoral factors that would neutralize both pathogens and toxins. Now, innate and adaptive immunity are inextricably linked, as was wisely recognized by the Nobel Prize Committee in 1908, who awarded the prize jointly in medicine to Metchnikoff and to Ehrlich. And indeed, in the intervening time, we have learned that products of the adaptive immune response, such as antibodies, can help the function of phagocytes through, for example, opsonization. And more recently, we have also come to appreciate the key role of the innate immune system in instructing the adaptive immune response. And that latter aspect is, in fact, largely mediated through the action of a phagocyte called the dendritic cell that was discovered by Ralph Steinman. And the key unifying concept that brought together innate and adaptive immunity is the one by Charlie Janeway, proposed in 1989, who envisaged that all microbes would possess microbial signatures that he called pathogen-associated molecular patterns that will be sensed by so-called pattern recognition receptors. And in the case of dendritic cells, it will be the signaling of these receptors that would promote the activation of the cells and their transition into a state in which they will be competent to prime T cells, in other words, generate adaptive immunity. Now, I want today to talk about dendritic cells, and in fact, I want to talk about one particular subtype of dendritic cells. We now appreciate that this is a family that's composed of multiple subtypes, but the subtype that's depicted in blue in this slide is effectively one that is marked across both mouse and man by high expression of a chemokine receptor called XCR1 and by a C-type lectin receptor called DNGR1, also known as CLEC9A. Now, these cells are interesting amongst dendritic cell subtypes, not only for being a discrete subset, but for having some interesting properties, such as the one depicted in this movie here, where you can see these unlabeled mouse DNGR1 high, XCR1 positive dendritic cells interacting with dead or dying cells that are labeled in red in this movie. And you can see that the dendritic cells move into these clusters of dead or dying cells, and in a very short period acquire a significant amount of the dead cell material, which they then transport with them. And I'm telling you this because it turns out that DNGR1 is not just a useful marker for these cells, but it is, as I've mentioned, a receptor, is a member of the C-type lectin receptor superfamily. But interestingly, it is not a pattern recognition receptor in the sense that it doesn't appear to have a PAMP ligand, rather that it is a receptor that allows dendritic cells to detect the presence of dead cells. And that can be exemplified, for example, in this experiment where we have taken the C-type lectin-like domain of DNGR1, in other words, the ligand binding domain, and we have used it to stain cells that are dead, that are permeable to this dye topro, and you can see on the right-hand side they're stainable with a CTLD of DNGR1, but not that of a control receptor called dectin-1. And the reason why DNGR1 is able to detect dead cells is because it recognizes a ubiquitous component of all cells that is exclusively present inside cells, and it is only therefore exposed when cells lose membrane integrity at the point of cell death. And that component turns out to be filamentous actin, or F-actin. And in this slide, you can see some in vitro polymerized F-actin forming filaments or bundles of filaments that are stained on the right with the extracellular domain, the ECD of mouse DNGR1, and on the left with an F-actin probe called a phalloidin. And we can detect this binding also by doing some detailed analysis, such as using biolayer interferometry, where, as depicted on the bottom left, we can anchor actin filaments onto a probe, and then we can dip that probe into a solution that contains the extracellular domain of DNGR1, which, as depicted at the bottom, is in fact a homodimer. And you can see on the upper left that you've got rapid binding, and then when you move the probe into a solution that doesn't contain DNGR1, you also see rapid loss of binding, and that translates into an apparent affinity of about 2 micromolar, which is quite respectable as far as actin-binding proteins go, but in fact it's rather modest if you consider the affinities of antibodies binding to their targets. But it's important to realize how this experiment was actually done, which is depicted here. You have the filament on a probe. You come in with the receptor. It binds, and when you wash it off, it comes off and is effectively washed away from the experiment. But as I mentioned, this is actually a receptor. That means that it's anchored onto a membrane. In fact, it turns out that it is a surface receptor on dendritic cells, so this could be the plasma membrane. But it is also an endocytic receptor, so that could also be the membrane of an endosome, and the recognition of the actin filament would occur either on the extracellular milieu or in the lumen of the endosome. And in these circumstances, if a few receptors were to lose binding, because the ligand would still be anchored by adjacent receptors, the probability of rebinding is much higher. And so we can try and mimic this process by setting up the experiment differently, by putting the receptor on the probe and then coming in with the actin filaments, which in fact turns out to be relatively difficult to do for technical reasons, and we have to use very short actin filaments for this. But when eventually we manage to do it, what we see is something quite interesting, which is you still have relatively fast binding, but most importantly, now the receptor doesn't come off the ligand, and your apparent affinity has effectively gone up by 3 orders of magnitude, and it is now in the nanomolar range. And this is, I think, a nice example of how avidity can play a key role in innate immune recognition, in this case, innate immune recognition of dead cells. Now, we can actually get greater detail into this interaction by looking at the complex of the NGR1 and F-actin, the structure of which was solved in collaboration with Keiichi Numbers Group in Osaka. And in the middle panel, you see the actin filament, which is 2 protofilaments wound around each other, decorated in blue with the ligand binding domains of the NGR1, a single C-type lectin-like domain. And in the reconstruction, the schematic on the right, you can appreciate that the NGR1 binds right at the interface between the 2 protofilaments, making contact with 3 actin subunits, and it beautifully explains why the receptor can only recognize a polymeric ligand, because that binding site does not exist in monomeric actin. And you can actually zoom into this interaction and look precisely at what residues on the receptor are making contact with actin, and you can in fact look at the key dominant residues, mutate them, and assess their contribution to the receptor binding. Now, you might be wondering at this stage why all of this is actually interesting or important. And the reason I think this is interesting and important actually comes from an experiment that has been done by many people over the years. This is our version of it. Which is that if you take dead cells that contain a foreign antigen and you inject them into an experimental animal, you can actually induce an immune response to those sterile cells. In this case, how you assess this immune response is by showing that the mouse is now immune to subsequent challenge with a tumor that expresses the same model antigen, which is ovalbumin in this particular experiment. And that tells us that sterile dead cells can be immunogenic. And this is because these cells can generate, for example, a cytotoxic T-cell response against ovalbumin, which we can quantitate, and we can show that that response is partially dependent on DNGR1 because it is decreased in DNGR1 knockout mice. So a simple explanation as to why this response should be decreased is simply that the receptor serves to take up dead cell debris by the dendritic cells, as in the video that I showed you earlier. But in fact, that does not appear to be the case because the DNGR1 deficient dendritic cells are just as good as the wild-type counterparts at taking up dead cells. So that led us to begin to wonder whether we were dealing here with something that is slightly different from the model that I pointed out at the beginning of my talk, even though it very often gets confused with the PAMP model by Janeway. This is actually a model that was independently proposed by Walter Land and Élie Metchnikoff. Walter Land actually coined the term DAMP for damage-associated molecular pattern, and their proposal was that rather than having PAMPs activate dendritic cells, maybe the contents of cells which are innocuous when inside cells, if exposed and come into contact with dendritic cells, for example, after cell necrosis, they could trigger this process of dendritic cell activation and allow for the initiation of a T cell response, which in this case could be in a sterile situation. And this could potentially be important, for example, in the case of anti-tumor immunity. Now, we spent quite a lot of time addressing this possibility, and to just summarize a lot of data in one cartoon, that does not appear to be the case. It doesn't appear to be that DNGR1 acts to activate dendritic cells in response to contact with dead cells. So effectively, we have a conundrum. We have a receptor that is dispensable for the activation of dendritic cells in response to dead cells. It is dispensable for the uptake of dead cell debris, and yet in knockout mice, we see a decrease in the effector T cell response, specifically to immunization with dead cell-associated antigen. So that led us to wonder what was going on, and one clue came from the realization that the effector T cell response that was primarily affected here was the CD8 T cell response, and effectively we focused our attention on this concept of cross-presentation, this mysterious process by which dendritic cells can extract antigens from a dead cell, probably through routing through specialized endocytic compartments, and generate peptides from that antigen for presentation on MHC class 1. And we don't really have formal evidence that this is the case, but I will just show you one tentative piece of data that indicates that actually something interesting about the routing of cargo from dead cells within endosomes is regulated by DNGR1. And here's that piece of data where if we look at dead cell debris in blue being internalized by dendritic cells, this is perhaps most obvious in this overlay here, you find that quite a bit of those dead cell debris co-localize with DNGR1 and with markers of the recycling endocytic pathway, such as RAB11, but in DNGR1 deficient mice, that co-localization is effectively lost. And so we don't fully understand what is going on, but this is the summary of what we believe, that somehow in response to contact with F-actin exposed by dead cell corpses, DNGR1 can alter endocytic traffic of those endosomes containing those dead cell debris in such a way that antigenic material from those corpses can be salvaged for presentation on MHC class 1. And so ironically, we've come back full circle to Metchnikoff, because what we're dealing with here is effectively the process of phagocytosis and endocytosis and the subsequent handling of the endosomal cargo. And so that seems to be an appropriate place on which to end and acknowledge the members of my group, and in particular those circled here that are recent contributors that have been involved in our analysis of DNGR1 and its biology.
Thank you very much, Caetano. That was a really great talk. So now we're going to move on to Ajay Chawla, who will be talking about the role of phagocytes in homeostasis and specifically in the regulation of thermogenesis. Ajay, thank you so much for joining us, and go ahead.
Thank you. What I'd like to talk to you today about is how the innate immune system participates in the regulation of body temperature, which is by a process called thermogenesis. So as a way of introduction, as Caetano just nicely shared with you, the classical view of innate immunity has been that it functions to sense pathogens and then initiate responses to eliminate infections both directly and via the activation of the adaptive immune system. But over the last few years, work from my laboratory and those of others who have been studying macrophages, mast cells, Tregs, and innate lymphoid cells suggest perhaps that the immune system has a much broader role in regulation of tissue homeostasis. In this extended view of immunity, what I'd like to suggest to you is that the innate immune system can function to directly sense environmental factors and then initiate responses that allow the organism to adapt to environmental challenges. And my lab has been exploring this hypothesis in the context of adaptation and acclimatization to cold environments, which mammals are able to do. So in response to sensing of cold, the brain activates the sympathetic nervous system, which will turn on what is called pre-existing thermal effectors to allow you to adapt. This includes things like shivering to generate heat and vasoconstriction to conserve heat. In addition to this, mammals have another tissue which can engage in thermogenesis, and this is called brown adipose tissue. Here, the sympathetic nerves release norepinephrine, which can then initiate a signaling cascade to activate allosterically a protein called uncoupling protein 1, or UCP1. UCP1 dissipates a mitochondrial proton gradient and thereby necessitates an increase in metabolic respiration, and a byproduct of that increase in metabolic respiration is the generation of heat. Now, these physiological programs work really well for the short term, but long-term acclimatization to cold requires the recruitment of new thermal effectors, which have in turn beige fat. Now, this tissue can be found in humans. For example, on the left, you can see in an FDG PET scan the uptake of the trace of glucose in the supraclavicular area, the para-aortic area, and near the perirenal regions. And if you were to biopsy these tissues, what investigators have found is that essentially they contain UCP1 expressing beige adipocytes interspersed among white adipose tissue. Now, this acclimatization process can be modeled in rodents, where you switch the housing temperature of a mouse from a temperature that's thermoneutral, in which the mouse does not activate any adaptive thermogenesis, to a temperature that's 5 degrees Celsius, let's say, for 48 hours. And what one observes is that the tissue, subcutaneous white adipose tissue, goes from energy-storing tissue to an energy-burning tissue on the right side. Now, what I want to introduce you to the concept is that this requires basically a relay of information and how information is passed on in metabolic homeostasis. So there are at least 3 different mechanisms. The first one is that hormones that are secreted in the circulation can get to target tissues. And my analogy here is the tissues are connected by roads, or the conduits being the vasculature. The second is the nervous system, which includes both the central nervous system and its outputs, for example, the vagal nerve and the sympathetic nervous system, and these allow for rapid activation of, for example, the cold setting of preformed thermal effectors. And the analogy here is Ethernet cable that relays information rapidly, allows you to communicate. And what I'd like to suggest to you today that there's a third means of communication, and that means of communication is enacted by innate immune cells, by their inherent ability to really go to every place in the body and thereby providing a signal. This would be analogous to the Wi-Fi system that we're used to using anywhere when we are not hardwired. So the question my lab has been studying is, how does cold, which is the only stimulus that activates thermogenesis, result in the recruitment of beige fat? Now, the canonical view has been that cold will activate the sympathetic nervous system, and this will then instruct the precursor cells that reside in the adipose tissue to convert into this thermogenic beige fat tissue. What we found was that in addition to the sympathetic nerves, there are signals provided by innate immune cells in the form of cytokines and neurotransmitters that regulate the transformation or the remodeling of this energy-storing tissue into a thermogenic tissue. And what I'd like to do next is to share with you some data in support of this model. So I'm going to focus on a type 2 cytokine signaling pathway, which involves the cytokines interleukin-4 and 13. They signal through the IL-4 receptor alpha subunit, the downstream transcription factor that mediates a response to STAT6. The basic experimental paradigm is mouse are housed at 30 degrees, which is thermoneutral, or in the cold, which is thermal stress. And you can see the tissue remodels into this thermogenic beige fat. But if a mouse lacks these type 2 cytokines, this remodeling response is abrogated. Now, this goes through the canonical signaling pathway because, as shown here, so on the left, you're looking at immunoblots. And essentially, as you put the mouse in the cold, you can see the induction of UCP1 protein. This is absent in the mice that lack the signaling receptor IL-4 receptor alpha. The brown adipose tissue, which resides between the scapula of the mouse, is hardwired by the sympathetic nerves, and there is no effect in this area. On the right, you're looking at the ability of the mouse to burn oxygen. This is like analogous to an athlete's VO2 or oxygen consumption rate. And what we observe is in the blue is a wild-type mouse as you shift the temperature from 22 to 4 degrees, it increases its thermogenic rate. But the one that lacks these IL-4 receptor alpha signaling pathways, it is reduced by 10 to 15%. It's a capacity for thermogenesis. So based on these observations, we were interested in 3 key questions. What are the cell types that make these type 2 cytokines to stimulate beige fat development? How does signaling via this receptor promote the development of beige adipose tissue? And can we bypass cold altogether and use the signal of interleukin-4 to promote thermogenesis? So we used a mouse called Forget, in which all IL-4 producing cells are tagged by GFP, and we found that in the cold, the dominant cell type that is competent for the production of an interleukin-4 is in the eosinophil. Now, if you took a mouse that lacked eosinophils, what we found was its ability to induce the expression of UCP1 or induce beige fat biogenesis was attenuated. And these mice, like the other mice I showed you, had impaired thermogenesis in the cold, as shown here in the pink. Basically, they had decreased oxygen consumption rate by about 15 to 18%. The next question we asked was, how does signaling by interleukin-4 regulate the biology or the recruitment of beige adipocytes? Based on the literature and our own data, we postulated 3 models. The first one being when the signal is released, it's seen by thermally differentiated cells, and it induces their transdifferentiation into beige adipocytes. Now, we found that this model was unlikely to account for what we observed in our studies. The second is that when the signal is released, it is seen by precursor cells, which will then favor the commitment of beige adipose to the beige adipocyte lineage. And the third is it's seen by an accessory cell such as a macrophage, who would then transmit signals in trance to regulate beige fat biogenesis. So for my talk today, I'm just going to focus on the third model, although we have investigated the second model also. When the mice were put in the cold, we found that they recruited Ly6C high monocytes in a CCR2-dependent manner, and CCR2 chemokine receptor was required also for beige fat biogenesis. But once the cells got there, they actually needed to see the signal via the receptor interleukin-4 receptor alpha, and some data to support that is shown here. So in the control animals, you can see the nice increase in UCP1, and as you delete the receptor in the myeloid cells, the ability of these mice to increase UCP1 is lost. Then in accordance with this, essentially these animals have a decrease in energy expenditure in mild thermal stress or severe thermal stress. So in my Wi-Fi analogy, the macrophages, which are the router, are simply not there, or what are there, but they're not connected to the internet and cannot relay the information anymore. The next question was, what signals are made by macrophages to support thermogenesis? In a previous study, we had discovered that the type 2 cytokines can induce a catecholamine synthesis biosynthesis pathway in the animals, and that's shown here on the right. And the key step in the biogenesis of catecholamine is the first step carried out by tyrosine hydroxylase, which is the rate-limiting step. So we partnered up with Richard Palmerter to ask the question, what happens if we delete tyrosine hydroxylase in myeloid cells? And what we found was that the loss of tyrosine hydroxylase, as shown here on the left slide of the slide, causes the deletion of TH in myeloid cells, reduces the total TH expression by about 80 to 85%. As a consequence, the steady-state catecholamine levels goes down by about 70% in these animals, and these animals are unable to induce UCP1 in their subcutaneous white adipose tissue in response to cold. There is some residual activity still there. That could be because the deletion is not complete, or because the sympathetic nerves are also present in the tissue. But the main point I want to make is that if you look at the TH protein expression in the whole tissue, one observes that the major adrenergic tone to this tissue is provided by the myeloid cells or macrophages. So in my Wi-Fi analogy, the macrophage, the router is still there, it's connected to the internet via the interleukin-4 receptor, but it cannot transmit the signal anymore, that being the catecholamines. And to briefly address the last, these mice, just as I mentioned earlier to you, have a decreased capacity for thermogenesis in the cold, as shown here. So to briefly address the last question, we asked, is this signal sufficient to induce the thermogenic tissue? And the way this experiment is done is shown here at the bottom. Mice are housed at thermoneutrality to turn off all programs of thermogenesis. We simply administer the cytokine interleukin-4 for 8 days and then do the analysis. And histologically, one can see that the tissue in the wild-type animals undergoes beiging or browning, whereas in knockout animals that lack the signaling receptor alpha, this response is completely lost. And if you just focus on the first two panels here, you can see the treatment of wild-type animals with interleukin-4 induces UCP1 and TH expression, but not in the knockout animals, suggesting that this is sufficient to drive phage fat recruitment. And probably one of my favorite slides for this vignette is that these animals actually have a higher capacity for thermogenesis when they're put in the cold. So this is shown here. You can see the blue trace is the control animals retrieve the vehicle, whereas the orange trace is one that received interleukin-4. Thermoneutrality or 30 degrees, there is no difference in thermogenesis, but the animals that received interleukin-4 have more thermogenic capacity and are better able to adapt to environmental cold, as shown here. And this is completely an on-target effect because it requires a receptor, that is the alpha receptor alpha. So what I hope I've shown you today is that we have started to deconvolute a circuit that controls the biogenesis of beige fat. We found that the group of innate immune cells, including eosinophils and macrophages, were required for this biogenesis. And in this context of cold exposure, eosinophils are the dominant cell types that make interleukin-4. The signal is genetically required in the myeloid compartment, in particular the macrophages, where we found a genetic requirement for tyrosine hydroxylase in the beige fat biogenesis. And these are some of the other papers that have found a similar pathway is also important in the recruitment of beige fat biogenesis in different contexts, suggesting this is probably a core mechanism by which the animal remodels in response to environmental cold. So I just want to thank the people. This study I shared with you was carried out by a former postdoctoral fellow, Yifu Chu, in the laboratory, and our collaborators were Rich Loxley and Rachel Palmerter for these studies, and I'm grateful for the grant support for the work I shared with you.
So now we're going to turn to our final presentation from David Russell, who's going to discuss the relationship between the host macrophages and microbacteria. David, thank you for joining us.
Thanks. Yes, so I'm going to talk about infection and the role of macrophages. Now, I mean, when I think about macrophages, I basically think of them much the same way you think about lavatory cleaner, because they will kill 99.9% of all known household germs. They do an extremely good job as a frontline defense against microbial invaders. So it's very interesting just how they're able to handle the pathogens. But because that interaction is an interaction that's a very frequent one, you can imagine how some pathogens have evolved to actually deal with macrophages. And Mycobacterium tuberculosis is perhaps one of the best examples in terms of a very, very long-term stable interaction between a pathogen and its host cell. So what I want to talk about are really three short perspectives, and that's to stress the intimacy of this relationship between the infectious organism and the macrophage as a host cell. And so first I'd like to talk how the bacterium responds to macrophage-mediated stresses. Secondly is looking at the impact of the macrophage on treatment with our current drug regimen for tuberculosis. And then thirdly is wrapping it all together to show why it's very important to include the macrophage or the host environment in drug discovery programs moving forwards. What we're looking at here is proteolytic degradation inside the phagolysosome, inside a macrophage. And it basically shows that much of the biology of this cell is translated through this intracellular compartment. And the green fluor that you see is basically generated when the fluorogenic substrate is cleaved by a proteolytic enzyme. So that's the environment that any pathogen has to deal with if it wants to parasitize this cell. And what Mycobacterium actually does is it has found a way to interfere with the normal maturation of the phagolysosome. So if you look at an IgG bead phagosome, so this is a phagosome containing an inert antibody-coated particle, you can see that it acidifies down to about pH 5 within 10 to 12 minutes. But Mycobacterium has found a way of blocking this normal acidification process, and the bug hangs out in a vacuole at about pH 6.7. So it's already starting to modulate the function of the host cell to promote its ability to hang around, to infect that cell. There's no doubt that TB essentially senses the environment inside the host cell, and it senses the host stresses. And many of these stresses are immune modulated. So, for example, superoxide burst. The production of superoxide radicals is increased if the host cell is activated with gamma interferon or with TNF-alpha. The pH of the vacuole, the bug, blocks the normal maturation process, but if the macrophage is activated, the macrophage actually overcomes that blockage and delivers the bacteria into a low pH compartment. There's acquisition of lysosomal hydrolases, there's production of reactive nitrogen intermediates, and there's nutritional restriction. So the bacterium has to deal with a wide range of host-derived stresses in order to maintain the infection. So what we've been doing to actually look at this is we've been doing transcriptional profiling of bacteria inside the host cell. And we've been identifying genes that are upregulated in response to specific host-derived stresses. And Xiumin Tan, who's a postdoc in the lab, who's just about to leave to a faculty position at Tufts, she's built these reporter strains of bacteria that express fluorochromes when they're exposed to different environmental stimuli. And so I'm going to show you how one of these works. So the one I'm going to look at is a reporter strain of TB that actually turns green when you expose it to nitric oxide. So it expresses M. cherry, a red fluorescent protein, constitutively, but when you expose it to nitric oxide, it upregulates the expression of GFP. And you can look at this inside infected mice. So this is lung tissue from mice that were infected 14 days previously with the reporter strains. And we're looking at naive mice and mice that we have vaccinated with heat-killed bacteria. So this already has a pre-existing acquired immune response to the bacterium. And you can strip away the actin, and you can, beg your pardon, you can strip away the actin. So you can strip away the nuclei, and then you're left with the bacteria. And you can see on the left-hand side in the naive mice, there's very little induction of GFP, and on the right-hand side in the vaccinated mouse at 14 days, there's higher level of expression of GFP. And you can take this out to 28 days, again, strip away the actin, strip away the host nuclei, and now you can see that there is almost equal induction under both conditions. Now you can score this where we're actually scoring the green signal from individual bacteria and then generate these scatter plots. And so you can see that if you look at the naive mice on the left at day 14, this is the level of expression of GFP versus the vaccinated mice at day 14. So there's a strong induction of GFP expression. By day 28, essentially, you've got that acquired immune response fully set up. And now there's very little difference between the vaccinated and naive animals. In the next slide, what we're doing is we're taking these reporter strains and we're now staining the tissue with markers of host response. And so this is stained with an antibody against the inducible nitric oxide synthase. And this is the enzyme that actually generates the nitric oxide in the tissue. And again, you can strip away the nuclei, and you can see that on the left-hand side, there's very little expression of the inducible nitric oxide synthase, and there's very little triggering of expression of GFP in the bacteria. But on the right-hand side, you can see strong staining in the vaccinated mice, and this correlates with induction of GFP. And again, day 28, when your bacterial response is much more comparable under both conditions, you can see, again, you've got expression of the inducible nitric oxide synthase in both of them. Now, moving on from that, I'm now going to look at a replication reporter, because the bottom line, what you really want to know about your bacteria is their replication status. And so here you can see there's a green dot. Every time the bug divides, it has a green dot. And that green dot persists for 70% of the replication cycle of the bacteria. So you actually can use this to score rates of replication in the tissue. And this is just looking, again, scoring rates of replication of the bacteria in the tissue. So looking at the wild type naive mice, you can see 40% of the bacterial population have green dots. If you look in the vaccinated mouse that has a pre-existing immune response, only 5% of the bacteria have green dots. And then if you look in the gamma interferon knockout mouse, 65% of the bacteria have green dots. So in this way, you can actually get a feel for bacterial replication in situ at the sites of infection in the mouse. So what we've been doing more recently, and this is work done by Lu Huang in the lab, is we've been basically taking the infected lungs, digesting them, and making single-cell suspensions. And the reason we've been doing this is that so that we can actually look at the distribution of bacteria within the phagocytes in the lung. And what you find is the bacteria are distributed in different subsets of phagocytes. So if you look at the 2-week panel, you can see interstitial macrophages, dendritic cells, these are monocyte-derived dendritic cells, and alveolar macrophages. And these are all the different cell types in the lung tissue that contain the bacterium. So what we're doing moving forwards with this is trying to understand which of the phagocyte subsets support bacterial growth and which of the phagocyte subsets control bacterial growth. So we can actually get a mechanistic understanding of the infection looking at the replication status of the bacteria. Now, we can look more deeply at phenotypes in these single-cell suspensions. And one of the phenotypes we've been interested in is how the immune status impacts the effectiveness of anti-TB drug treatment. So we've taken this single-cell suspension and we've sorted the cells using a flow sorter into CD80 high or activated myeloid cells and CD80 low or resting myeloid cells. And these are myeloid cells that contain live TB, and they're isolated from the mouse lung. And this is work that was done by Yanchen Liu in the lab. We then put those cells in culture and we exposed them to either isoniazid or rifampicin. And what we found was the bacteria in activated macrophage actually have higher tolerance to drugs than the ones in resting macrophages. So the advent or the emergence of the immune response actually drives acquisition of drug tolerance in the infecting organisms. And if you look at this drug tolerance in a bit more depth, what you find is if you look in mice that actually are defective in the inducible nitric oxide synthase, you do not see induction of drug tolerance. But we think that nitric oxide is, in the mouse system, nitric oxide is one of the main drivers of the acquisition of drug tolerance. And then moving on from that, thinking more widely about drug development, and this is work that was done by Brian van der Ven, who's now a faculty member at Cornell. And what we did was we screened compounds for compounds that would kill TB inside a host cell. And we screened 340,000 compounds. And we used fluorescent bacteria, and this just shows the screening platform in the BL3, and we looked at the ability of compounds to block the expansion of fluorescence or the growth of the bacteria. And the really neat thing was when we got to the end of the screen, we'd essentially got about 300 high-potency hits. And those high-potency hits we could divide into 2 classes. They were constitutively active, which are the compounds that look like the antibiotics we have out there now. And there were conditionally actives. And the conditionally actives only worked when the bacteria was inside the host cell. So we would never have found these compounds if we screened in rich medium. So we've been working on these conditionally active compounds to try to find out what they're acting on. And what we found is that 75 of the 101 conditionally active compounds actually hit cholesterol metabolism. So Mycobacterium tuberculosis is quite unusual in that it's able to degrade cholesterol, which is not a common phenomenon for bacteria. So we've hit several different pathways in the cholesterol degradation pathway. And you can actually see that the bacterium inside the host relies on cholesterol and fatty acids as its primary carbon source. And it actually modulates the macrophage physiology to render those carbon sources more available. So if you look on the left two panels, these are macrophages that we've fed with oleate, with oleic acid, to drive this foam cell formation. But you can see after 48 hours, they've lost these lipid droplets. But if the macrophages are infected with TB, they retain the lipid droplets. So TB actually modulates macrophage metabolism and turns it into a foam cell, a foamy macrophage. And you can see on the right-hand panel that the bacterium themselves have little lipid inclusions inside them. So the bacterium have changed the physiology of the host cell to enhance their access to nutrients to allow them to grow better. So taking all of those things together, what I hope to have persuaded you is that in TB infection, the unit of infection, we really should think of this as the infected macrophage and not the bacterium in isolation. Because the environment within the macrophage impacts TB physiology, TB infection modulates macrophage physiology, and this interplay reveals new conditional drug targets and impacts the activity of known frontline drugs. So moving forwards, we really need to interrogate the bacterium within the context of its host cell. And so just to round off, I'd like to thank National Institutes of Health and Bill and Melinda Gates Foundation for the financial support of the work that I've discussed. Thank you.
Thank you very much for the talk, and to Ajay and Caetano as well. We do have a handful of questions, and in the remaining time, I and Alil would like to go through some of these. The first question is for Caetano. Caetano, we have a question from postdoc Ravi Kant at the India Institute of Science, and he's wondering whether, if I can understand this question properly, have you identified the sequences, the amino acid sequences in F-actin that are actually binding to DNGR1, and perhaps these sequences can be found lodged onto MHC class 1 molecules in the process of cross-presentation?
We can identify the residues in F-actin that we believe are the key interactors with the residues that we have identified on the receptor. Proving those by mutagenesis is much trickier because you will alter the structure of F-actin to the point where the experiments become uninterpretable. As to whether these residues would be preferentially presented by MHC class 1, I mean, obviously, each MHC class 1 molecule is going to have a particular motif that it binds. So other things being equal, one might have thought that things that are associated with F-actin or F-actin itself could preferentially generate epitopes for presentation. But that really would depend on the specificity of the peptide binding motifs of the particular MHC class 1 molecules that one is dealing with.
In theory, would it be possible to biochemically purify MHC class 1, do acid elution followed by mass spec analysis to see whether you can find particular peptide sequences from F-actin eluting from the groove?
Yes, I mean, that would certainly be possible, but the problem, of course, is that the dendritic cells themselves express plenty of actin, and so if there is an epitope that is particularly has a particularly high affinity for the particular MHC class 1 molecule that one is interrogating, you would not be able to discriminate whether that came from material ingested by the dendritic cell or by the dendritic cell itself. I think what we can try and do to address the spirit of that question is an experiment that we are currently trying, which is to ask it slightly differently, which is, if there are antigens preferentially associated with F-actin, will those be preferentially cross-presented? So, for example, we can try and make a version of albumin that would have a peptide that allows it to bind non-covalently to F-actin, and now ask whether that improves the efficiency of cross-presentation. And that is an experiment for which we don't really have an answer yet.
Okay. Thanks for that.
Okay, thank you. Yeah. So the next question is for Ajay. The question is from Amit Singh at NIH in Sousa. The question is, what is the phenotype of these mice once they can't generate a cold response, and how they respond to infections, for example, when they have a fever?
The first part is straightforward in the sense that the animals, when they lose about 10% of their total thermogenic capacity, drop their body temperature by 1 to 1.5 degrees by something called a Q10 effect for all enzymatic reactions. The second part of the question that Amit asked, sorry, can you repeat the second part of the question? Can you repeat
they have a fever?
Fever is a different then defense. So cold exposure results in defense of your body temperature. Fever is the set point at which the body is maintaining its temperature is shifted upwards. And the type of response or the innate response that regulates fever, at least, or initiates it, is type 1 immunity in response to, for example, bacteria or viruses. So it's a very different thing compared to normal thermogenesis in the cold.
Okay, thanks for explaining that.
David, we have a question from a faculty member at the University of California in Irvine, Charita Madiraju. And the question is, the compounds that target cholesterol metabolism, is there any specific target of cholesterol? Have any studies been done to characterize the primary biological target of the hits identified from this screen? Do these hits identified on the screen have any... I'm sorry, David, go ahead.
So the short answer is yes. The problem is, of course, there's a large number of compounds. So we've been working through these. For some of the targets, we've actually identified the enzymes that are hit. So, for example, we have inhibitors of PAMPS, which is involved in propionate detoxification, which is a downstream activity, because digestion of cholesterol generates a large amount of propenyl-CoA, and the bug actually doesn't like propenyl-CoA. We've got inhibitors of HSA-B, which is an enzyme complex that's involved in opening sterol rings in cholesterol. We've also got some inhibitors or some compounds that modulate an adenylate cyclase and generate high concentrations of cyclic AMP. And under those conditions, the bacterium behaves as though it can't see cholesterol at all. So there's a wide range of targets. We don't have them all defined, but we do have some very specific modes of action that we do understand. So it's a range of answers.
Cholesterol itself is critically important for signaling of receptors at the plasma membrane of cells. So I'm wondering whether you're seeing any toxic effect on the host macrophage if you're going to be impinging on the ability of the macrophage to transmit signals.
I think that essentially you're operating in a cholesterol replete environment, so I don't think it's going to impact on that kind of function. What we see most markedly in the TB-infected macrophages is that cholesterol efflux is shut down, and the ABC transporters that are most actively involved in the release of cholesterol from the cell, their activity is really closed down. So it's a retention phenotype, I think, more than anything else.
Okay. Okay, so now I have a question. You have more?
No, go ahead, Alil.
Yes. Okay, so I have a question for Caetano. So the question is, if dead cells are immunogenic, would not normal turnover, let's say, for example, in the gut, cause inflammation?
Yes, so I mean, these are important questions. So the first thing that one has to realize is that obviously a cell will only be immunogenic provided it has an antigen against which you have a T-cell repertoire. And so if a dendritic cell is constantly sampling dead cells and presenting the antigens from those dead cells, and there is no foreign antigen present, it's effectively presenting self, and it's no different from the dendritic cell presenting its own antigens, because there's effectively no real repertoire to speak of to respond to that. So I don't think that in most circumstances there will be any danger of eliciting, for example, an autoimmune response. Now, the other thing that I should make clear is that in most cases where there's natural tissue turnover and natural generation of dead cells, these are effectively apoptotic cells that even if they get ingested by dendritic cells, will largely not have lost membrane integrity at the point of ingestion, and therefore F-actin will not be released, and therefore the NGR1 will not be engaged. So this particular mechanism that we are describing likely evolved to detect the presence of cytopathic events such as cytopathic infection or perhaps necrotic centers of tumors, which would signify pathological circumstances and which would often be associated with the presence of a foreign antigen. And that's where you would want to favor this cross-presentation.
I see. So they would also recognize injured cells, for example, if they were broken up.
Pardon? Say that again?
So the dendritic cells would also recognize injured cells that were broken up, but they wouldn't recognize just normally apoptotic cells.
That is correct. As far as we can tell, this receptor is only engaged when fragments of cells are created where F-actin is exposed or intact necrotic cells. Now, it can be a secondary consequence of apoptosis. If apoptotic cells are not cleared sufficiently rapidly, then they do undergo membrane disintegration events and F-actin becomes exposed. In fact, the cells that were used in one of the experiments I showed in the slide have exactly undergone that type of secondary necrotic disintegration. But in most cases, that would not happen with a normal load of apoptotic cells and a normal rate of clearance.
Okay, thanks for clarifying. Okay.
Ajay, I have a question from... Yes, Ajay, I have a question from a listener at the University of Ulm in Germany. And they're asking if you have identified the myeloid subset or the macrophage subset that's actually driving the transdifferentiation of white adipose tissue to beige adipose tissue.
Thanks for the question. I want to clarify one point up front. We don't think transdifferentiation is a major pathway, and this is consistent with findings of others in the field. So the type of macrophage or monocyte that drives this response, it seems to be that the tissue is thermally stressed, recruits lysic C-high monocytes, which are classically thought to be inflammatory. But once these cells get into the tissue, they actually see the signals of the type 2 cytokines 4 and 13, which allows them to undergo alternative activation, or sometimes referred to as M2 activation. And that actually is the trigger for the cells to gain competence for the production of catecholamines and perhaps and make other factors that are necessary for the growth of beige fat.
Interesting. I'm going to ask for moderator privileges to ask a question that's been fascinating me, Ajay, which is, and forgive my naivete, but I generally think of temperature as a function of the migration, or excuse me, the vibration of atoms. So if you have a lot of kinetic energy of atoms, you have heat, and if you don't have a lot of kinetic energy, you have cold. So that's why it's cold in outer space, because outer space is a vacuum. So if that naive sense is somewhat correct, then what is it about the lack of vibration of atoms that causes eosinophils to secrete IL-4? Bizarre question.
Okay, it's probably beyond my reach right now, but let me just give it a shot, Bruce. So I think one thing that's underappreciated when we talk about thermoregulation is the fact that metabolism. We think of metabolism as making ATP to carry out all the cellular processes. But metabolism, just like a combustible car engine, is not efficient. Roughly, if you were to burn a mole of glucose or through enzymatic machinery in ox-phos versus combust a mole of glucose, you'll find that the energy stored in glucose, we can only harness about 50% of it. The rest, 50%, is essentially being escaped as heat, which would contribute to, for example, kinetic energy of a system. Now, with regards to eosinophils, producing interleukin-4 in response to cold, we don't think this is. We don't think the eosinophils or the macrophages per se are actually directly sensing cold. We think that cold is being sensed by peripheral sensors that are probably neuronally encoded, and that responses are somehow integrated in the brain. Those nuclei that do this integration haven't been identified yet. And then there's an efferent response that actually activates this innate immune response. And what that efferent response is unclear also. But it would be something analogous to a circuit that Kevin Tracy has identified involving the vagal nerve innervating, the spleen to have an anti-inflammatory response becoming activated during sepsis. It would be some sort of and then that would be an analogous way to think about it.
Mm-hmm. Fascinating. Alil, do you want to wrap up?
Yeah, well, I have a final question for David. This is from Natalie Frank at the Scripps Research Institute. The question is, does what you have just described for mycobacteria also apply to other intracellular pathogens, as well as perhaps other pathogens, bacteria, fungi, viruses? And in your view, should all pathogens be studied in the context of the host cell?
Okay, so I'll take the last point first, because I absolutely believe that is the case, that whether we're doing vaccine development or whether we're doing drug discovery, that we really need to incorporate the environment of the infection into the screen, because otherwise I think you're throwing away a whole lot of potential targets. So I think it's very important to have a biological screen that is rich as it possibly can be and has as much context from the host as you can possibly generate. So fortunately with intracellular pathogens, that's a little bit more easy. But I prefer to actually screen inside the macrophage rather than just drop the pH of the medium or think that we can mimic that environment, because I basically assumed a long time ago that I wasn't clever enough to do that, and it was much more effective to just screen inside the host cell. In terms of relevance to other pathogens, the questions are going to be the same for each pathogen. So the challenges that face each pathogen is the same, but there's no doubt that different pathogens have dealt with these pressures in different ways. And some pathogens escape into the cytoplasm and so avoid the whole endosome-lysosome process. Other pathogens actually are capable of surviving within the acidic lysosome. So I think different pathogens have definitely evolved different routes, and some of them will be similar, but I think in fact more of them are different than the same.
All right. Well, thank you very much. So, although there are many more questions we could ask, we have run out of time. So I think we have to wrap up now. So thank you so much, David, and also I would like to thank all of our speakers, Caetano Reis, Sousa, Ajay Chawla, David Russell, for all their great presentations today. I also want to say we're very grateful to our sponsor, R&D Systems, for their contributions that made this webinar possible. And thanks for tuning in today to learn about the biology of phagocytes. If you missed anything during this webinar or you'd like to listen to it again, there will be a recording that will be made available shortly on the Cell Press website. Also, if you have any comments about this webinar or any suggestions for the future, we'd love to hear from you, and you can email us at cpwebinars@cell.com. Do keep your eyes out for future CellPress webinars and symposia, and don't forget to check out the upcoming Cell Symposium, 100 Years of Phagocytes. You can find all the information about this and other future programs on our website at cell.com/symposia. So thanks everyone for listening. We hope you enjoyed this webinar, and we do hope you will consider joining us again in the future. Have a great day.